So Richter syndrome is a very severe complication in indolent lymphomas, particularly in chronic lymphocytic leukemia patients. You see in a few patients that they progress from this rather low malignant phenotype towards an aggressive lymphoma. And this is a very severe clinical problem since these patients rapidly progress due to the aggressive nature of the lymphoma. But also due to the disease being very refractory to our current chemotherapeutics and also targeted drugs that are not very effective in these patients. And the only viable option that we have right now would be allogeneic stem cell transplantation, but otherwise these patients have only very limited options, and we have a very urgent need for further clinical trials and novel substances and combinations in Richter syndrome.
And what causes that progression from one state to the next?
So for the time being, we know from genetic investigations that have been done in Richter syndrome patient samples that there are a couple of mutations that occur up front, like the p53 mutation, notch mutation that has been described by several groups. But there is also a group of patients that do not have a specific genetic aberration or a clear driver mutation that explains why they progress from CLL towards Richter's transformation. The difference in our approach is that we are not screening for genetic aberrations. We applied a hypothesis-driven approach because we have seen that AKT plays a pivotal role in CLL. We know that it is downstream of the B-cell receptor signaling pathway, and downstream of it a couple of very important regulatory mechanisms, particularly the PI3 kinase AKT pathway, are how a cell proliferates, how a cell regulates its own homeostasis, energy levels, translation, and the resistance towards programmed cell death or apoptosis. So we were wondering if that specific pathway is involved in progression of CLL. And we applied different approaches. The first one was that we investigated samples from Richter syndrome patients and looked for the activation of AKT, which would be a phosphorylation at a certain amino acid residue that is really crucial for the activation of AKT. And we could see in these samples that we have phosphorylation of AKT in Richter syndrome samples, while we don't see it to that extent in normal CLL samples. And the second approach, which actually really led us to the observation that this might induce Richter syndrome, was that Thomas Wunderlich generated a model in a mouse where we constitutively activate AKT by tagging a specific site to this protein, which directs it to the cell membrane, and that actually mediates that this protein is constantly activated in cells where we express this construct. And we made this as a conditional allele, so we could actually activate it only in specific cells in a mouse. So we crossed it to certain strains that activated those constitutively active AKT by CD19-CRE only in B cells. And that we crossed to a chronic lymphocytic leukemia model. And so they could see that mice that were bearing the chronic lymphocytic leukemia allele TCL1 and the constitutively active AKT, that these mice developed Richter syndrome. And in these mice, what you see is that they have a significantly reduced lifespan, that their appearance of leukemia drastically changes. So when you look at the morphology of the cells that you observe in the bloodstream, they become much bigger, they have a transformed phenotype, they look like transformed blastoid lymphocytes. And additionally, they have much larger spleens, and in these spleens you see that the architecture is totally disrupted, and they have a higher frequency of mitosis, and you see very large cells, blastoid cells, and this corresponds to the phenotype of a diffuse large B-cell lymphoma. And by definition, this is a progression from chronic lymphocytic leukemia towards a diffuse large B-cell lymphoma, which is a Richter syndrome. So this is now the opportunity to model Richter syndrome in a mouse, which we did not have for the time being. And this also opens up novel perspectives to use this model as a preclinical platform for treatment of Richter syndrome.
When it comes to that treatment of Richter syndrome, would you count then AKT to be at least a biomarker or possibly a target for targeted molecules?
Yes, that could potentially be an option for future clinical trials in Richter syndrome patients. So we see that a certain subset of Richter syndrome patients has phosphorylation of AKT when we stain this with immunohistochemistry, and this particular subset of patients could be a particular target for future clinical trials when we actively would target this pathway either with upstream or directly AKT targeting compounds.